My thanks to ARM for allowing me to moderate this session today. I'm going to let my panelists introduce themselves. So Claudia, why don't we start with you at the end?
All right. I'm Claudia Mitchell, Senior VP of Product and Portfolio Strategy at Astellas Pharma, and former CEO and co-founder of Universal Cells, a company that Astellas acquired a couple years ago.
And I'm Matt Kane, co-founder and CEO of Precision Biosciences, a leading genome editing company with focus areas built really around allogeneic CAR T and in vivo gene corrections.
And Pascal Touchon, I'm the president and CEO of ATARA Biotherapeutics, a leading T-cell allogeneic off-the-shelf immunotherapy company dealing in cancer but also in autoimmune disease like multiple sclerosis.
Good morning, everyone. Sam Kulkarni, I'm the CEO of CRISPR Therapeutics, and we're focused on both rare diseases and immuno-oncology, and in immuno-oncology mainly with an allogeneic CAR T approach.
Great. So guys, to warm up, the session today we're going to focus primarily, as you can see from the title, Emerging Therapies for Cancer. We're going to be looking at T cells and related products. You've already got a flavor of this rich panel of experiences, all driven by the common theme that we all want to transform the practice of medicines with the modalities we're developing. But Pascal, if I can start off with you, I think you and I are very lucky guys that we saw the birth of Kymriah, the commercialization, initial inroads with that product and that platform. As you look back now, what are the key lessons that you're drawing as you now take on the helm of ATARA and applying the lessons learned from those times that we spent together?
Yeah, I think it's been a real journey towards bringing the first gene and cell therapy to the approval stage in the FDA, with the FDA, and then globally across the world. And of course, we always have to thank the patients and the investigators and the scientists at UPenn that helped this come true, but also us starting the journey, me continuing, and now somebody else following that. But the most important is when you think about it, just in a few years of time, these particular products, then others products have been approved since then, have been approved not only in the US, but also in Europe, in Australia, in Canada, in Japan. There's been regulatory approval in different indications. There's been pricing and reimbursement approval. And you've seen the slide that Janet was presenting there. This is amazing that within a short period of time, these transformative therapies that are really trying to change the life of patients with very severe type of leukemia and lymphoma have been approved, even though they are very new in the way the CMC part is being put together, the manufacturing, but of course all the clinical part as well. They've been approved by the stringent regulatory authorities, and they have been approved by pricing and reimbursement authorities. So this has been an amazing journey, and I think we should all be pleased with that, to see that as a way to move forward for the field, that when you bring truly transformative therapies for patients, there is a way to get approval, there is a way to get pricing and reimbursement and access for patients. Now, this journey has been having some bump as well, so it's not been a straightforward road. And these bumps have been very significant in many aspects. There have been bumps in terms of development, in terms of manufacturing, both the process development as well as the capacity of manufacturing for autologous CAR Ts, which is always a challenge there. And then there have been bumps in terms of access for patients. Many, I would say, in countries where it was such a challenging situation to bring these new therapies that probably the payers' environment was not fully ready to integrate that. So when we think about Kymriah, for example, whether it was around having outcome-based agreement, when you are in countries where you have a system that does not really allow to have a price zero, if you have no benefit of the treatment, is a challenge. The fact that you have countries like in the US where when you have an hospitalized Medicare patient, you have a diagnostics-related group, a DRG system that does not move very rapidly, then that's a challenge because then hospitals do not receive the full refund for the treatment, even though there is the need to cover these patients fully. So this has created some barrier to access, and I think that's certainly something we need to work as an industry and with ARM and others to make sure that we worked more on anticipating the arrival of these new therapies, these transformative therapies, so they get full access for the patients there.
So Matt, a fantastic busy year 2019 for Precision Biosciences, building out the company, building out manufacturing. And as you look at the lessons learned from 19, what do you reflect upon as you look at the initial commercializations of particularly the cell therapy space?
Yeah, well, I think kind of building on what Pascal just said, as a backdrop, I think what's been so impressive has been the uptake of these new therapies. given all of these pressures and access issues and the like. And we say, why is that? And it's the response rates you're seeing with patients at late stage that have progressed from all the other therapies. And that's what makes this field so exciting. And so if we can now address these issues of accessibility and the rest with some new innovative approaches, it really brings, I think, a great deal of enthusiasm to the space right now. I think that's why we're all here today, and why we're so optimistic that going into 2020 we have some a new set of questions to start to answer and unravel in terms of how we can make these products more accessible.
And Claudia, as I look, busy year 2019, Universal T cells and Astellas, and then looking to further transactions. Clearly, in your new home of Astellas, you've been watching this space, but what are the key lessons internally you guys are debating, particularly with your role as mastering the strategy and portfolio in this direction? What are you observing? What have you seen that's making you think about how you're going to be developing your businesses in the future?
Well, at Astellas, we really focus on the allogeneic cancer cell therapies, and we think that because pricing and costs and manufacturing are key issues in this space, and we think that having allogeneic products will really help decrease the cost and make the supply chain much simpler, and have the really improve the accessibility of these treatments. So we think that we can take the lessons learned from the autologous therapies and try to build therapies that are even better and improved by making allogeneic cell therapies. And we focus both in T cells with a partnership with Adaptimmune, but we also are developing NK cells. And we think that the first products are autologous T cell therapies, but I think that the cell therapies for cancers are going to be expanded way beyond that. We are going to have other cell types. We are going to have cells that are engineered in many, many ways. And so this is just a start.
So Claudia, you teed it up beautifully for me to turn to Sam now. As I think about 2019, what really caught your eye as you thought about the progress in the science? Clearly, leading CRISPR Therapeutics, one of the first vanguard companies in this space from a CRISPR platform perspective. Amazing progress happening in activities with Vertex for you guys. What really caught your eye in terms of giving you that renewed energy and confidence in this sector around the prospects for allogeneic therapies as Claudia was teeing up?
Thanks, Oz. Just to put it in context, before I talk about 2019, is if I think about the last 100 years, we are privileged to be in this time where we're coming up with these new therapies. 100 years ago in cancer, it was all about surgery. How do you, what kind of surgeries you want to do, how you take cancers out. Then we had an era of very toxic chemo agents, all the way from wartime agents into more advanced agents to try to kill these cancers. And that took years, decades almost. Then we had an era where antibodies really showed hope, and then some targeted agents. But we're now entering a phase of cell therapies. And I would argue today that cell therapies are here to stay in cancer. We can talk about all the challenges of commercialization, of autologous, and kudos to you guys who were there at the start. Cell therapies, I think, will be a big part of cancer medicine. I would argue that in liquid tumors, in the next 5-6 years, we're probably going to see at least a third of the market be cell therapies in liquid tumors, whether it's autologous or allogeneic. And overall, I think in cancer therapy, once we make a dent in solid tumors, there's no bounds to what cell therapies can do. For us, really, in 2019 at CRISPR Therapeutics was a remarkable year because we disclosed our first data in sickle cell and beta thalassemia. And it's these patients who were walking around with years of disease and requiring a lot of transfusions, requiring a lot of hospitalizations, all of a sudden getting a one-time treatment and essentially getting a functional cure to this date, at least it's early data. But that just shows that in cell therapies, you can make these cells, you can administer them as these are live cells that you're putting into a patient. In fact, the manufacturing scale-up's happening in the body. And so you can actually put these in patients, make sure that it doesn't do anything that you don't want it to do. And there are a lot of questions to be answered on how efficacious they're going to be, how do you engineer them to be better. But this is the start of what I think could be very revolutionary. So we're quite excited at CRISPR Therapeutics for the whole field.
And Matt, your landscape 19, what really caught your eyes in terms of real movement of the needle in the allogeneic thinking, particularly from your lens from precision?
Yeah, well, I think we got to answer a few of the initial key questions that the allogeneic CAR T field has faced. And one Sam touched on just briefly, but it was this question of, are we going to see severe GVHD by giving foreign cells to the patient? And I think now with several studies, including data that we presented, I think at least at the dose levels that we've seen these products be utilized, that has not so far been a concern. So it's really encouraging for the field. It tells us that at least in the ways that we're using these products thus far, this might not be something that stops us. The other thing I think we started to learn this year that was a concern, a real question for the field is how hard or how far back would we have to push a patient's immune system so that these cells could at least persist long enough to show some evidence of cell killing, because there had been a strong argument in the field that we might have to push those patients' immune system back so far that could put the patient in a very precarious position. And I think we're starting to see some data now that's giving us some real hope that that's not going to be the case. And this is important because I think the goal that we all have with allogeneic cell therapy products is, again, all about accessibility and giving physicians the ability to utilize these products in the widest variety of ways possible to really meet the patients where they are and be able to address their diseases appropriately in the position that they find them in. And so that really gave us a lot of encouragement last year, and as we're going into 2020, is really that part of the reason we're so excited about the prospects for this field. To Sam's point, it may take a number of iterations before we're at a point where we feel really good about these products, but certainly no doors have been shut for us as we start going down this path, which is at this stage what makes it so encouraging.
Pascal, from your perspective, as you look at the needle moving in terms of allogeneics, clearly that's an area close to your interest.
Maybe if I think about 2019, what really caught my eyes was ATARA Biotherapeutics, because I joined them.
Well, I was going to
6 months
say,
ago, so.
Obviously the diligence panned out, right?
It is very
them, yeah.
personal, but no. And I think more importantly, I think what is interesting is to see the development of allogeneic T-cell immunotherapy. And as you know, ATARA Biotherapeutics has a product to treat a very rare type of lymphoma called PTLD that is now in phase 3. And having an allogeneic T-cell immunotherapy in phase 3 is something that is very important for the field. And we've announced that we plan to initiate a BLA filing in the second half of 2020. And not only this is important to see development and a kind of regulatory goal there, but beyond that, we are delivering that product today in our clinical trials and in compassionate use within 3 days, just the shipping time from inventory to the patients. And I think that's really what we're here for in the allogeneic field, which is to be able to deliver a product from inventory to patients within just a few days. So having the ability to do that and doing that every day today in the field with compassionate use, with our phase 3 program, with our new programs as well that we're developing for this product, and doing that not only in the US, but outside of the US as well, this is a true achievement for the field.
And as we think of allogeneic therapies as it relates to immuno-oncology and cancer, we're looking at various cell types. We're talking about iPSC cells, NK cells, universal T cells. So Claudia, I'm going to turn to you in terms of those emerging trends that you're seeing. I mean, even macrophages now. So how are you synthesizing all of this, and what are you thinking in terms of these cell types and the pros and cons, if you may just touch on that for us?
Yeah, sure. I think that, as I said initially, I think that T cells are just the beginning. I think that there is a lot of hope and potential for the the immune cells to be used as well. For example, you mentioned macrophages. There are companies starting to develop CAR macrophages, and those cells are different than T cells in the way that they not only can phagocyte the tumor cell, but they can also they are antigen-presenting cells, so they can stimulate the immune system and fight tumors in different ways. So you also have NK CARs and NK cells that are engineered to fight tumors. And those cells act in a different way. They can be used also to enhance monoclonal antibodies actions. Each of these cells you have an arsenal of immune cells that you can deploy in different ways, and each one will have a specific what mode of action. And so I think that depending on the tumor, depending on what you want to do you want more persistence, do you want less persistence? I think that we're starting to build a much more complex panoply of things that you can use, and I think that that's very exciting. I think that the field is, I fully agree, the field will just grow, and the possibilities of modifying those cells to do exactly what you want is really a very robust way of doing it. For example, and in our case at Astellas, we start our starting material are iPS cells, so you can gene edit those cells and then differentiate them into NK cells or T cells or macrophages, whatever you want, and so you have a batch of cells that is completely characterized, and you know exactly you can do the gene modifications the way you want beforehand, and you have a batch that you can indefinitely expand. So I think that there are many flavors of cell therapies that are just arising, and it's a very exciting time for the field, I think.
So as you guys know, my company, Tmunity, is focused on T-cell engineered products, CARs, TCRs, with a focus on solid tumors. We are an autologous-based company that's now in research stage for allogeneic as well. So one of the challenges that I think it'd be great to debate today would be about persistence and durability of allogeneic products. We've seen, I think, a real shift in the thesis there in terms of the early days where these products were viewed very much as transitory in the early days with allogeneics. I think the needle's moved, but I'd love to get your perspective as you now look at the field and you think of cancer as a whole. Because persistence and durability, repeat infusions for solid tumors, this is going to have to be figured out. So I'd love to get your perspectives on how you guys are looking at this individually in your own companies, particularly with the editing in mind, the cell source in mind, thinking about how do you get these cells to really persist with constitutive expressions of interleukins or other payloads. Just give us a feel of how you're looking at the landscape as you think about evolution past hematological malignancies. Maybe start with Sam.
Yeah, thanks. It's a really important question. The key thing we're trying to do is increase durability of response, right? That's the objective function. And that actually is a two-part equation. It's how hard you hit the cancer and how long you can have these cells do their work. So it's not just about persistence, it's also the initial response. In fact, the analogy is in some of the surgeries, you know, if you're able to carve out the margins very cleanly, cancers don't come back, regardless of the adjuvant therapy. But if you don't have clean margins in surgery, the cancers come back. So the hope we have is that with allogeneic CAR-Ts, that you actually hit the cancers really hard, because you're coming up with these CAR-Ts that are coming from a healthy young person who has a very robust immune system. And we've seen in labs that they're much more cytotoxic than cells from a patient with a weakened immune system. Right? So one, you're hitting the cancers pretty hard. Second is then the persistence question. You can argue that you can engineer these cells, have different subtypes, the memory subtypes that last longer. You can engineer these cells for persistence by making them more stealth, and it's one of the strategies we're undertaking by knocking out the beta-2M gene, and many more such strategies. Or we can also multi-dose with these allogeneic CAR-Ts since they're off the shelf. So it gives us many degrees of freedom to achieve what's ultimately the objective function, which is increase the durability of response. So I see these as early stages in the persistence argument. There's a lot to do, and I'm actually very happy that we're taking divergent approaches to how to increase persistence, and time will tell what the best strategy is.
Pascal? If I may.
Yeah, I think there are two aspects in your question there. One is, do we know exactly how long these cells should persist to have durable effect? We don't know. And it might be very different from one type of pathology to another one, maybe very different even within liquid tumors, and of course in solid tumors. So when you don't have yet the data there, you have more to look at what is really important, which is to achieve this durable response. And I believe personally, and we believe at ATARA, that persistence is a key there, especially if you build a platform. Because if you build a platform, you want to have the possibility for your technology to address liquid tumors, to address solid tumors, to address allogeneic CAR-Ts, allogeneic TCR-Ts, allogeneic T-cells themselves. So whatever the type of target, to be able to have a similar type of platform and technology that allows the cells to persist. And we've seen persistence with our healthy donors type of origin cells that are being developed as TAP cell or other type of product we're developing as allogeneic T cells, that we have that persistence. We have that persistence, and that was very interesting, even in patients that are not immunocompromised. Because, of course, if you lymphodeplete the patient, by definition, you could have more persistence there. But the idea was to say, can we have a situation where you still have persistence without lymphodepleting the patient? And we've seen that in nasopharyngeal carcinoma, for example, with tuberculous cell. We've seen persistent up to 23 months of some cells. So that's really, we believe, the way forward, where you build a platform with allogeneic T cells where you can have persistence, but of course, the impact of persistence will be depending on the different type of patients. And then you have retreatment, as you say, which is also a very important aspect of allogeneic T cells. So we have experience now with tuberculous cell of retreating because we do 2 to 3 cycles, and each cycle is made of 3 infusions. So we have experience of treating patients repeatedly with allogeneic T cells without having any safety or even efficacy impact there. So that's really how we should build this platform, the ability to be extremely flexible in addressing different needs, because today we don't know yet what is a true relationship between persistence and durability of response in specific disease.
Matt, I'd love to get your thoughts as well. Same question, please.
And I will provide an equally unclear answer because it is a very complex issue, but one in which we think is important. Again, to Sam's point, it's durability that we're after, and there's a variety of ways we could potentially get to a durable response. And without question, persistence is part of the equation. But then we have to ask, well, what time point are we actually looking at here? Are we talking days, months, years? And then what sort of cells do we want to persist? Does it help us if these cells are lingering around for a long time, but they're completely exhausted? I mean, what are they doing? So again, the depth of response piece of it is also really important. It's something that we're really focusing on at Precision, in part, again, because we do have at least the potential to repeat administer this product, which makes it a lot more like a traditional biologic in the traditional ways that one goes about utilizing these types of therapies. So it is, again, I agree, it's a question that we're going to have to wrestle through. And again, to make the point again that one of the things we're so happy about in this field is that everybody is trying something different. And as data comes out, we're learning from each other, we're getting better at this, and ultimately, my hope and my real belief is that we're going to find better ways to serve the patient and be able to again, give physicians more alternatives that can help address the real needs that the patients have, because the requirement for, say, persistence may be very different from one patient with one disease to the next. And we're only going to really fully understand these things over time. And it's going to take a variety of different approaches for us to really figure out which one of these variables is critical for us to get over the hump for different diseases.
So, Claudia, I'm going to switch now the controversial question, starting with you at the other end. Allogeneic's going to replace autologous therapies.
Absolutely.
Care to elaborate?
Well, I think that for a few reasons, and I'm not saying that there won't be any use for autologous in the future. I think that, and broadly speaking, allogeneic will be the mainstream because first of all, the manufacturing is much simpler and cheaper. So that is a big point. The other point is the availability of these cells is much more, they're readily available. It's much, so for example, for certain cases, like if you're talking about ALL, where some patients currently cannot get the treatment because they just cannot wait for the therapies. If you have a readily available source of allogeneic off-the-shelf cells, you can treat those patients. So that's one other aspect. And yeah, so in our case, we have iPS cell-derived cell therapies, and these are indefinite sources of cells for the patients, and you can manipulate them in vitro beforehand. You can have a batch that is completely consistent. You can really characterize your cell very, very well, and you can use that for all patients. And so you not only, it's cheaper to manufacturing, you have much better clinical outcomes, because you don't have batch-to-batch variations. You control your product much better. So I think that, yes, they will eventually replace for most cases.
So maybe I have a slightly different context to Claudia when it comes to sort of the ease of manufacturing. And I think you're right, the scalability and the future vision is there. But I personally believe that there is still a long way to go in terms of figuring out, particularly the cell source manipulations, and very importantly, I think both autologous and allogeneics are going to be plagued with one challenge, which is the back-end analytics, which is really the driver of cost and time when it comes to our businesses. And so leading on to the manufacturing equation here, so as we think about this, Matt, from a precision biosciences perspective, right, you and I kindred spirits in terms of manufacturing investments. We were, as a seed stage company, really proactive in acquiring our own manufacturing facility and building that out, both for viral vector and also hopefully in the future for cell therapies. You again have opted for a build out. So no one model is perfect, but walk us through the thinking as you were thinking of the scale up, capacity needs, CDMO versus internal build. Where are your heads at now as you've gone through that journey?
Yeah, no, I think it's a really important question in this space. I mean, manufacturing we've learned, is absolutely critical. And I think we're very fortunate now, strategically, to be able to have our own manufacturing facility. Now, I'll also say, though, we continue to work with a third-party manufacturer, and it gives us some additional flexibility. I mean, the advantages with a third party are that we were able to more quickly produce clinical material prior to our facility being ready. We were able to leverage expertise from our third-party vendor, and it didn't require that upfront investment in capital to actually get into the clinic and start serving the patients. But it's extremely expensive, and there's not a lot of bandwidth in our industry right now for external providers. One of the advantages we have now with our own facility is that we control the supply. We control the staff. We have more control over our highly proprietary process. And we've seen a dramatic drop in our cost of goods. So all of those things, I think, long term, made it a pretty easy decision for us to invest in our own facility, and I'm glad we had. But again, continuing to have the flexibility of a third party to pick up additional programs and the like, I think is also a really nice thing to have going forward, and we expect to continue to be working with third parties in a variety of fronts.
So Pascal, I mean, ATARA, massive investments over the past decade, right, since the inception of the company from a manufacturing perspective, but clearly one size doesn't fit all. How do you think about, as you think about scale and build for your pipeline, very ambitious pipeline?
Yeah, I think just to add to, I agree with Matt, and to add to that, there's maybe 3 additional advantages of having the control of your own manufacturing. One is that what we're doing at ATARA is to collocate together, really at very close proximity, a few feet, the people from translational science, from preclinical research, from process science, from quality, regulatory, and manufacturing. Yeah. Because they have to talk every day about what's happening in cell therapy and how do you improve that. And one evidence of this working is that over this year, last year, 2019, sorry, we managed to increase very significantly the yield of our process manufacturing. So we started the year at the beginning of 19, from one leukopak, one apheresis, we could make 120 doses. We finished the year where we can make 400 doses for one leukopak. So that's really this type of work, this proximity of the different people working together to be able to do high-quality manufacturing in increasing the yield. And that's where we believe that at a commercial stage, we will be at a cost of goods that will be very similar to biologic today. So that's something very important. And I think the third aspect is the ability to leverage a particular unit to make different type of product. So all you need today is able to make not only allo T cells like TAB cells, but allo CAR Ts or allo TCR-Ts. The same unit, the same people, doing different things in a modular way. So that's also offered by this type of internal manufacturing. So that's an investment. It's capital investment, of course, but it's investment that is really extremely interesting and important to create profitability later on for this type of companies.
Yeah, and we've seen the value from an M&A perspective for those companies that had the vision in the early days to establish manufacturing, vis-a-vis Avexis and Audentes. And Sam, you have a very unique relationship with Vertex. Vertex also on the M&A front, recently acquired Semma, my good friend Bastiano Sana's company. You and I used to work together as well in a former life. And clearly you have optionality as well as you think of CRISPR's pipeline. So how do you guys factor in the CDMO versus internal organic build equation?
Yeah, I think for us, the answer is a mix. I think there's no one way or the other. You know, we started obviously with more CDMO support, and now we're building more in-house. I think there's the benefits of owning your manufacturing are very clear, right? Flexibility, quality, ability to innovate, keep the IP. But at the same time, I think with CDMOs, you get, capital efficiency. You're able to multi-source. You're able to risk mitigate on certain fronts. And there's actually real value add that comes from our CDMOs from a science perspective and product development perspective. So what we have is a bit of a mix, and I see the evolution in a little bit of the auto space. Back in the day, all the car companies used to make every component themselves, and it just wasn't very capital efficient. And then it became a point where they would source a lot of different raw materials, and they would do the assembly, which was the key part of building these cars. And now, obviously, Tesla has changed the paradigm completely in a different way, different direction. But for us, we don't need to make all the guides. We don't need to make all the Cas9 enzyme. I think we can source it. There's many different sources of it. And having a multi-source approach actually reduces the risk. But then the actual cell assembly, the cell manufacturing is something where there's still a lot of innovation happening. It's not in the days where you had research and development and you hand it off to manufacturing and it was a different silo, right? I think it's much more of a continuum as you go from R&D into manufacturing, and there's so much innovation happening. So proximity, that sort of intertwined nature of innovating is very important. So we're making a lot of investments in building our own cell therapy manufacturing, of course, with capital efficiency and other considerations in mind.
So we got about 8 minutes left. 2 areas that I want to touch base with you guys. All of us are involved in gene editing in one form or another in our companies. Each of us have our biases and the platforms that we focus on, but love to get your very brief synopsis of pros and cons in the approaches that you're taking or evaluating potentially if you're able to disclose that. So Claudia, maybe I could start with you in terms of the views of Astellas as you think about the world of gene editing and modalities and how you apply them.
All right, so at Astellas we have our proprietary gene editing platform based on AAV gene editing that was acquired via the acquisition of Universal Cells. And we use that to not only manipulate to engineer the HLA gene so that the cells are not recognized by the immune system, but we also can insert and gene edit any other gene in the genome. So it gives us a lot of flexibility. The drawback of our approach is that we need to subclone the cells at each step. So it works for iPS cells and pluripotent stem cells that can be indefinitely expanded, but it doesn't work for a T cell, for example. We can't use this approach directly with a T cell, but so we go from iPS cells in order to do that. So in terms of, so I don't see a lot of cons in that approach because we're starting from iPS cells, and we think that there are other advantages of the expandability, notably, is a big advantage there. But in terms of, so the gene editing per se, I don't see the drawbacks. I might say that, for example, the drawback one of our products, which is an NK cell, is probably the persistence and being able to make sure that we have a long-lasting treatment. But the fact that we have cells that can be re-injected because they're not immunogenic, we can overcome that. But I think that gene editing offers a lot of benefits, so I don't see cons on that side.
And Matt, with Arcus and your platform?
Yeah, so within the, maybe to back up a bit, so Precision Biosciences is a bit of a unique genome editing company in that we utilize a technology called ARCUS. ARCUS was derived originally from an enzyme called ICRE1. And what ICRE1 does in nature is it doesn't just break DNA, but it's evolved to insert a small copy of its DNA into the host of a type of algae. And so we really leverage this feature within the world of allogeneic cell therapies to site-specifically insert the CAR into the TCR locus at very high efficiencies. And beyond that, we're the gene editing company in allo CAR T that does everything it can do to avoid genome editing. That's it. That's the only editing step we do. Anything else we're trying to do going forward, at least with our current plan, is to utilize other tools and avoid using additional edits, which is a different strategy than many of our peers are using. We're doing this to avoid things like translocations and really to optimize the cell phenotype and the yield that we get at the end of our manufacturing process. So intentionally, a different strategy, a different path than a number of our peers in this space, but we're able to do this through the use of this very unique genome editing platform. And again, a different approach that we'll be interested, obviously, to see how it compares with others in the field that are taking a different approach towards additional edits and additional technologies into the cells.
Thank you. Pascal?
Yeah, I think it's fascinating to see how many different technologies could lead to the same type of objective. So I think it will be great because we'll see many of these going to the clinic or further advancing the clinical stage, and that will tell us a lot about the differences of different approaches. What we're doing at ATARA Biotherapeutics is, again, very different from my colleagues, is to take T cells that we can take from healthy donors that are able to be very selective to EBV and to specific antigens of the Epstein-Barr virus there. And therefore, we don't need to modify these cells so much because their natural TCR is the one targeting EBV. And then we just add, when we make an allo CAR T to this particular cell, a CAR. So we try to minimize, like yourself, the genome editing there to make sure that we preserve the properties, the immuno properties of these cells. And these cells, the EBV T cells, are really amazing cells because they are very much immunoprivileged. They have a very nice balance of phenotype between memory cells and effector cells. And we know that they expand, that they persist, that they traffic to the tumors there. So we're trying to preserve this immunocompetency of these cells and do as limited genome editing. And we use a retrovirus just to insert a CAR there at this stage. And we are also now moving into another type of construct and we have a mesothelin CAR T going to the clinic this year, which is on top of having a CAR, we add a PD-1 DNR, which is a very interesting idea to have an intrinsic activity against the PD-1, PD-L1 axis there. So it will be in the clinic this year, so we'll see how it delivers, but we're very excited about it.
Sam, I think I know your bias, right? Pioneers in the CRISPR therapeutic space, but how you look at the pros and cons of the approaches, and the landscape's moved a lot.
All these platforms can actually do the edit, right? You can optimize them, you can engineer them, they'll do the edit. I think the key difference is actually what edits you choose to make and the editing strategies. Obviously, I evangelize CRISPR because it's relatively fast-out technology. I went into the lab and I was able to make an edit without much help, so it shows you how easy it is to apply. It's slightly easier in the manufacturing setting as well. But the technology is not going to be the differentiator. It's going to be the strategies applied, which edits you're making, what combination of edits. And over time, my suspicion is we're going to get to like 8, 10 edits in these cells as we learn more about these cell therapies and what they can do in cancer.
So coming to the end, guys, it's the start of a new decade. We all have visions for where we see this sector going in particular. My vision that this is a decade for meaningful hope for patients, real meaningful hope in terms of transformative therapies. What are you aspiring to see for this sector? Starting with you, Claudia, as a close.
I think that we'll see in the next decade a multiplication of cell types being used for cancer therapies. As I said, we're going to have a growth of the allogeneic space, hopefully several approvals of allogeneic cancer cell therapies, and really solid tumor cells, solid tumor cancers being treated by cell therapies. That will be a big thing too, and I think we are getting there. So So lots happening, a big growth in the space.
Matt?
Just looking at 2020, I'm highly optimistic that we will start seeing evidence of durable responses within the allogeneic cell therapy field. I think given the number of different approaches and different strategies that are being used, I think that's highly likely to occur sometime in 2020.
Pascal?
I think certainly agree that durable response with allogeneic will be seen in 2020. I think emergence of solid tumors, I think for me is really where the field can take a different path. I mean, there's been amazing years. It could be even better years for patients if we are able to have truly effective T-cell therapy for solid tumors there. I mean, Tmunity, ourselves, others are working on different approaches there. We need these approaches to deliver clinical results that shows that these cell therapy are effective in solid tumors. And I will add, and maybe again, an ATARA Biotherapeutics portfolio aspect, that we have a therapy that is an allogeneic T-cell therapy for multiple sclerosis. So it'd be nice for the field as well to see cell therapy, allogeneic T-cell therapy, able to show safety, of course, and we've shown that already, but more importantly, efficacy in autoimmune disease. And MS is a very exciting disease because there is a medical need in progressive MS, which is really very important. I mean, these patients are deteriorating day by day, and we need to do something about it.
And Sam?
Yeah, I agree with all the panelists. I think progress in solid tumors and then seeing more durable response in liquid tumors. But from a business standpoint, I think I expect to see two things. One is big pharma waking up to cell therapies and finally recognizing this is going to be mainstream in the future. And second, I think we're going to start seeing some winners and losers in space. We have a lot of different companies and approaches, but we'll start seeing what works and what doesn't.
So ladies and gentlemen, we've concluded this panel. Please help me thank my fellow panelists.